All right, so let's take a look for a moment at different types of infections. So we just looked at HIV and how its processes through the host cell, and it's a reverse transcriptase or a retrovirus. Now we're going to take a look at persistent infections. We're going to take a look at the two different types. So a persistent infection occurs when a virus is not completely cleared from the system of the host, but stays in certain tissues or organs of the infected person. So this wouldn't be a situation like we see with the bacteriophage, for example, like the lytic cycle, where it gets in, makes copies of itself, and bursts out of the cell, and then goes on its way. This is where we have a persistent infection. So over time, our body doesn't go in and clear our system of this virus. Instead, it stays in certain tissues or organs for a very long time. The virus may remain silent or undergo productive infection without seriously harming or killing the host. So the mechanisms of persistent infection may involve the regulation of the viral or host gene expression or the alteration of the host immune response. So we'll talk about that in just a moment in a little bit more detail. So there are two primary categories of persistent infection. So there's a latent infection and there is chronic infection. So examples of viruses that cause latent infections include the herpes simplex virus. So the herpes simplex virus is one, both the oral herpes simplex virus, which you know as a cold sore on the lips and mouth region, and then also genital herpes, which is a more long-term, as is noted here, latent infection in the genital region. Then there's also varicella zoster virus, which causes chickenpox, which lasts about 10 to 12 days. But then there's also shingles, which is more long-term. It's the latent piece of that then can show up later. And then there's also the Epstein-Barr virus, which causes mononucleosis, but then can hang around longer and stay in the body for many years and then can creep back up again. And the hepatitis C virus and HIV are two examples of viruses that then cause long-term chronic infections. So our latent infections examples are the herpes simplex virus, both oral and genital, the varicella zoster virus with chickenpox and shingles, and then the Epstein-Barr virus giving us mononucleosis and then what's called is walking mono. And then our long-term or chronic infections are hepatitis C virus and HIV. So let's take a look at the latent infection first. So again, not all of these animal viruses are going to go through this process of the lytic cycle. There are viruses that can remain hidden or dormant inside the cell, and that's what is called latency. So when it goes dormant, it's not causing a serious infection anymore. It's just kind of hanging out in the body. That's latency. So these types of viruses are known as latent viruses, and then may cause latent infections. So viruses that are capable of this may initially cause an acute infection and then it'll go dormant. So what we typically see is that when the person is first infected, then they show infection, there are symptoms of an acute infection, but then after that passes, then there's this latency and it goes dormant for a while. So for example, the varicella zoster virus infects many cells in the body and it causes chickenpox. Which if you're familiar with that, you have sores all over the body, rash of blisters that are covering the skin, but it only lasts about 10 to 12 days. And then 10 to 12 days after the infection started, then the disease resolves, so those go away, the person is feeling normal again, but the virus isn't gone. The virus just goes dormant, and then it stays living within these nerve cell ganglia for years. And then during this time, the virus isn't killing the nerve cells. It's not continuing to replicate. It's not causing chickenpox again. It's just hanging out in our nerve cells. It's not clear why this does this, but in some cases, after many years of dormancy, then the virus gets reactivated, and then it causes a new disease that's called shingles. So oftentimes this will happen in situations where the person becomes stressed. So what we see is we have this dormancy, and it's kind of similar to what we saw in the lysogenic cycle, where once situation gets a little bit difficult, the virus is going to then go through the process of the lytic cycle, make copies of itself and get out of the cell. It's not the same, but you can think of it that way, whereas something will then happen, something switches, perhaps a stressful situation occurs, something changes in a person's life, and then what can happen is then it reactivates that virus, and then we get a new disease called shingles in this case. Chickenpox affects many areas in the body, but shingles is a nerve cell specific disease, and then it emerges from that ganglia where the virus is dormant. So we see it in more localized areas. And that's what this image is here. So in A, we have the virus that causes chickenpox here. It's an enveloped icosahedral capsid. And then in B, it's just showing an example of what shingles looks like, a very painful, itchy rash. So latent viruses may remain dormant by existing as circular viral genome molecules outside of the host chromosome. So that's also a little bit different. We don't have the situation where it's a provirus within the chromosome. It can be, but they also see, or it's also seen that they can exist as a totally separate viral genome hanging out outside of the host chromosome, so not integrated. But others do become proviruses and integrate themselves into the host genome. So there are just those two differences that are seen. During dormancy, viruses do not cause any symptoms, and they may be difficult to detect. So you may not even be aware that you're carrying the virus at all. So a patient may be unaware that you're carrying the virus unless a specific viral diagnostic test has been performed. That's common with the Epstein-Barr virus. So once a person has mononucleosis, oftentimes when they're younger, then that Epstein-Barr virus goes dormant and then hangs out in the bloodstream. And then when things get a little bit stressful or something changes within the body, then that can then come back out and affect the body in a similar way to mononucleosis, but isn't considered mono at that point. Then we have our chronic infection, which is the other type of persistent infection. So our chronic infection is a disease with symptoms that are recurrent or persistent over a long time. So in this case, it doesn't go dormant. It's going to continue to cause issues, continue to cause persistent infection over time. So some viral infections can be chronic if the body is unable to eliminate the virus. So an example is HIV. So our body is unable to eliminate HIV, so it stays within the body, causing a chronic infection. So sometimes there is a period of latency, though. So we, again, can see this when the person first gets the infection causes many symptoms, becomes a problem, a person is ill, and then we see along a period of latency, but over time, this virus is still affecting the body. And we can see that once a person becomes infected, the virus can be detected in tissues continuously thereafter. It's constantly there. But sometimes patients don't have any symptoms for many years. But, however, the virus maintains chronic persistence through several mechanisms. And it maintains this chronic persistence because it does these several things to make sure that it stays in the body. So it interferes with immune function. It can prevent expression of viral antigens on the surface of infected cells, for example. So our body is pretty smart. We have some pretty smart immune system cells, for example, that when it identifies something that's not self, so for example, when it identifies a virus, then it will recognize that and it will send up a little flag saying, hey, I've seen this virus. Go track down other viruses that have this same protein on the outside of them and kill them. HIV, once it's inside of the cell, it infects the helper T cells, which are an immune system cell. It will actually prevent those cells from expressing those antigens, from putting that flag out and saying, hey guys, it looks like this, go get it. So it stops it from doing that. It also can alter the immune cells themselves, sometimes making it so the immune system cells just don't work as immune system cells. It doesn't kill them, but they're not actually functioning as immune system cells, so they're not killing other viruses. Also, it can restrict expression of the viral genes So it's basically, it commandeers, it hijacks all the machinery within the cell, and it can tell it what to do or not to do. So it may restrict expression of the viral genes, therefore not setting off any alarms to the immune system. And then the immune system just doesn't know that it's there. Also, it can rapidly change viral antigens through mutation. So viruses also are very smart, specifically HIV, in that it rapidly changes. It can change the way it looks. It can change the proteins that it expresses. It can change all of these things so that when the immune system picks up on the fact that it's there, it'll say, like I had mentioned, it'll put up that little flag saying, hey, it looks like this, go get this guy. And in the process of the body saying, hey, it looks like this, go get this guy, the virus has already mutated and already changed so that it doesn't look like that guy anymore. So then the immune system has to get hip to that and then go after that. And then by that time, it is perhaps already mutated again, and it keeps staying a step ahead of our immune system. So eventually, the damage to the immune system results in progression of the disease, leading to acquired immunodeficiency syndrome, which is AIDS. So that's when HIV has kind of taken over. It's no longer a latent kind of chronic infection. Now it has become AIDS. And the various mechanisms that HIV uses to avoid being cleared by the immune system are also used by others, like hepatitis C. So we see the same thing happening in hepatitis C, where it's a chronic infection. You can't ever get rid of hepatitis C, and it goes through similar changes, alterations, mutations, and things like that to maintain itself within the body to make sure that the body can't get rid of it. All right, so we're going to take a quick look here at the life cycle of viruses with plant hosts now. So we do have plant viruses. They do exist. They are more similar to animal viruses than they are to bacteriophages. So plant viruses may be enveloped or non-enveloped. Like many animal viruses, they can have either DNA or an RNA genome. They can be single-stranded or double-stranded. However, most plant viruses do not have a DNA genome. So most of them actually have the positive single-strand RNA genome. And so then, of course, that acts like the mRNA. It can just quickly go into translation and make proteins and make copies of itself. So plant viruses may have a narrow or a broad host range. So for example, they give the Citrus tristeza virus. Infects only a few plants of the citrus genus, but the cucumber mosaic virus infects thousands of different plants in various plant families. So in this example, oftentimes when we're talking about bacteriophages and viruses with animal hosts, we see a much smaller host range. In this example, with the plant hosts, we see a very broad host range, for example, with the cucumber mosaic virus. Most plant viruses are transmitted by contact between plants, so they're next to each other. Plants often are next to each other. They drop their seeds, and then a new plant grows right next to it, and so they can actually bump into each other when they're blowing in the wind and then transmit the virus to the plant next to it. Also, it can be transmitted by fungi, nematodes, insects, or other arthropods that act as mechanical vectors. So not only can they be transmitted from plant to plant just by knocking into each other, but then we have these other vectors that are mechanical vectors. Remember, a mechanical vector means that it's just stuck on that thing, and then it gets to the thing that will be infected, and then it infects it. However, some viruses can only be transferred by a very specific type of insect vector. For example, a particular virus might be in aphids, but then not in whiteflies. So there are viruses that are specific to a vector, might be transmitted, carried by aphids, but not by whiteflies. And then in some cases, viruses may also enter healthy plants through wounds. So when a person is pruning, then perhaps it might get a virus in it, or weather damage. If it gets broken by the weather, then it has a wound, and then it might get a virus that way. Viruses that infect plants are considered biotrophic parasites, which means they can establish an infection without killing a host. So they hang out. It's similar to the lysogenic life cycle. They can hang out and stay within the plant for a long time. Here they're saying it can be asymptomatic or latent, and then it can lead to cell death as well later in the lytic infection or the lytic infection. So the life cycle begins with the penetration of the virus, just like we've seen in the other examples. Then the virus is uncoated within the plasma or the cytoplasm of the cell. So that's when we take that outer coating off. We get rid of the capsid. Okay, we get rid of the envelope, we get rid of the capsid, and then that releases the genome into the cell. And then depending on that nucleic acid, then the cellular components are used to replicate that genome and synthesize the viral proteins, and then make our new virions or virions. So it goes through a very similar process that we've talked about with bacteriophages and then also with animal cells. To establish our systemic infection, then the virus must enter a part of the vascular system of the plant, such as the phloem. So the phloem is kind of this tubular system of cells that generally go in the stalk part of the plant. So in the trunk of the tree oftentimes, or the stem of the plant, and then goes through the major veins of the leaves and such. And it carries nutrients throughout the plant. So a virus, in order to establish this systemic infection, is then going to need to enter the phloem, for example, and then hang out within the phloem, which is a major part of the plant, and then can move around in the plant because that's what is used to move things like carbohydrates and things like that for the plant to live, the nutrients it needs. The time required for systemic infection can vary from a few days to a few weeks, depending on the virus, the plant species, and the environmental conditions. So the virus life cycle is then complete when it is transmitted from an infected plant to a healthy plant. So in a similar way, when we see it burst out and then go and infect other cells, like other bacteria cells or other eukaryotic cells, well, not eukaryotic, but animal cells, then we see that also here with plant cells, which would be eukaryotic as well. All right, and then I think lastly we're going to talk here about our viral growth curve. So it is unlike the growth curve for bacterial population. The growth curve for a virus, we have our initial stage, which is the inoculum of the virus, and that's when it causes infection. And then, let me see if I can scroll down here. Not quite. Let's finish up the word up here. So in the eclipse phase, the viruses bind and penetrate the cells. So when we're talking about the different phases, first we have the inoculation. So when we inoculate, that's when the virus first causes infection. Then we have the eclipse phase. That's when the viruses bind and penetrate the cells with no virions detected in the medium. Let's see here. Here we go. All right, so here's step one. We can look at the actual chart here. So here's an hours going this way, and then here's the number of infectious virions, or virions here. So step one, we have the inoculation. So the inoculum of the virus binds to the cells. So they're just going to attach to the outside of the cells. Then we have our eclipse phase here, step two. So we see the number of viruses here in the inoculation part because it's on the outside of the cell at this point. And then once it penetrates the cell during the eclipse portion, then we don't see a whole lot of movement as far as numbers of viruses because they're within the cells and they're going through the process of replicating their DNA, they're transcribing, they're translating, all of these things, and they're building all of their pieces and putting them together in order to make the virus. So we are not able to see very many of them at that point. Let's see here. Then we see a burst. So the next step is the burst. And the number of virions per bacterium released is described as the burst size. So what we see here is that we see them building, and then we have this burst part where the host cells are going to release all of these viral particles. So we see them increase, because we've now burst the cell open and released all of these virions into the exterior. And then up here is the burst size. Which is the highest this chart goes, which is the maximum number. So the number of virions released per bacterium. So in a one-step multiplication curve for bacteriophage, the host cells lyse, releasing many viral particles into the medium, which leads to a very steep rise in viral titer. So a viral titer is the number of virions or virions per unit volume. So looking at our test tube, this would be our solution, and then a titer would be how many virions we see per unit volume. So this would be measured in the number of virions or virions in a certain amount of fluid there. So if no viable host cells remain, then the viral particles begin to degrade during the decline of the culture. So if it's gone through and it's already infected all of the cells in the culture, then we would see a sharp decrease here, because then the virus would just die out because there's nothing to infect them, because remember, they cannot live on their own. They have to be within a cell in order to survive. All right, so I think that sums up 6.1 and 6.2, and then we're going to move into isolation, culture, and identification of viruses.